The purpose of this tutorial is to demonstrate how to run a Southern blot using the Huntington's disease case. So we're just assuming we've just double-clicked on the executable, we've opened up the case application, and I'll click on this button to begin. And then just continue on to the screen that appears, which is the data screen. And we need to acquire the DNA sequences. We'll click the DNA button on the silver button bar. Inside this folder is another folder called Cases, and we'll double-click on that. I'm going to double-click on the genetic disease cases, Huntington's disease, double-click on that, and what we want to do is the Southern blot example, so I'll double-click on that, and then I will use case A as an example, so we'll double-click on this one as well. Now you'll notice there are a series of DNA samples inside this particular folder, and the easiest way to open them is by shift-clicking. I'll click on the first one for the ant, and I will hold down the shift key to highlight all of those and then click open. And you'll notice that they have appeared in what's called the open in process window. Next, what we need is an enzyme to digest those DNA samples. If you go back to the silver button bar, there is an enzyme button. And I'll click on that. Bring this over so you can see it. Scroll down and double click. You can either click and open, or I'll just double click on the enzyme EcoR1. And that will appear here as a line in the open in process window. And the name of the enzyme appears in the upper left hand corner along with its cut site, GAATTC. In order to digest these DNA samples then, and before I do this I'm going to actually make this bigger. But I'm going to click on this again, make sure that enzyme is activated. Whatever enzyme's been activated will appear up here. In this case we only have a single enzyme. I will click on the ant's DNA sample and then hold down the shift key and highlight all of them so I can digest all of them at once. And now I will go over to the silver button bar where it says Cut DNA. Click on that. You will see several options, but in this case all we want to do is digest it with this single enzyme EcoR1. So I will click that button, and I'm going to make this even a little bigger. You can see that all of those have been digested, and I'm just going to temporarily drag this over so that you can see all of them have been digested, and you can tell by the blue color and by the name of the enzyme which of those are the digested samples. You can also tell by these 3 asterisks. So if you have 3 asterisks in front of the name, that's a new file that's been created that appears in the open in process window representing the digested file. Next, what we need is a probe sequence, and I will go back to the silver button bar, and here is the button that says probe. I click on that, and from the list, wherever it might happen to be, in this case at the bottom, I will double click on probe Huntington's disease, and you'll notice that that file appeared at the bottom of the list, and the actual sequence appeared in the upper left hand corner of the main screen. That's the probe we need for a Southern blot, and the purpose of this tutorial is to run a Southern blot as quickly as possible, not going through all the individual steps. So we will do that, and all we need to do now is to basically indicate which of these samples we want to run the blot on. So I will take the digested sample. The order may be different, but they should all be in this region of the open in process window. And in my case, the last digested sample is the uncle sample. So now we have the digested samples highlighted. We have the probe, and we know that probe is active. We don't have to click it again. We know it's active because it appears in this window. To do this quickly then, we'll use the quick load and run button. To Southern blot, and I'm going to load the wells and run the Southern blot in one step. There's another tutorial where you can see how to load wells in a more stepwise fashion, but we want to do things quickly. The default timer setting is 60 minutes. At that particular timer setting, these bands are really too close together to see what's going on. So we need to separate those bands. We really want to rerun this blot at a longer duration, so we can separate the bands, and we can do that quite quickly by changing the timer setting. Or double click to highlight that. I'm going to type in 150 minutes and click set. And to quickly rerun that blot, all I have to do is use the run button and go down to Southern Blot.
And now there's more separation of the bands to make it easier to analyze the case. Information can be added to the label underneath the blot, but it's easier just to move the loaded window into position and then save that image using the screen capture capabilities of your computer.
The purpose is to demonstrate what we're going to call the differential display feature of CaseIt. But we're going to assume that we've already run the Southern blot for a case, and you'll notice that all of these bands are of the same color intensity. In some situations, you might want to know how much DNA is present in a particular band. And to get a feel for that, we can go up to the differential display button and turn this feature on. And when you do that, you'll notice that some of these bands are somewhat darker in appearance. These four and these four here and these two bands are darker in appearance than the other bands, and that relates to how much DNA is present in that particular location. In addition, there are a few options with the differential display. You can use it either with gels or with blots, as we have done here. So we won't demonstrate the use with gels, but the same basic principle applies. And you'll notice you can change the minimum densities of the various bands. So if I select low, it's fairly faint. So in this particular case, it's probably best to go with the default. So I'll put it back to the high value, and that makes things just a little bit easier to see. The purpose of this tutorial is to run a dot blot for one of the scenarios of the breast cancer case. The dot blot will involve DNA samples, so I will click the DNA button on the silver button bar and go into the Cases folder, which is inside the CaseIt folder. Go inside the Cancer folder, select Breast Cancer, and we will select Case A. Now you'll notice right away there's more than one DNA sample. There's actually 6 samples. So regardless of the order, click on the first one and then find the last one and hold the shift key down and so that all 6 of those are highlighted, and then say open, and then those files will open in the open in process window. The next thing we have to do is to run PCR on these samples. To run PCR, we need a primer. Here is the primer button of the silver button bar. Scroll down to find that and open it. I'm going to make this a little bit bigger. To run PCR, we will click the first DNA file, hold down the shift key, and click the last one. And now I'm going to go down to this quick load and run button, and I'm going to say run PCR. These are all PCR products. There are 6 of them, and you can tell they're PCR products because PCR is in parentheses and there is an arrow at the beginning of every one of those lines. The next thing we need to run a dot blot is a probe file. I will go to the probe button on the silver button bar. I'm going to go down and find the first probe and scroll down to find the last probe. Hold the shift key down so all 6 of those probes are highlighted, as you can see, and then I will click the open button. Here are the 6 original DNA samples, the primer file that we used to generate PCR products for those 6 original DNA samples, and then 6 different probes. We're going to have 6 different columns. All of these will be in one column associated with probe 185, then we'll put them in the second column associated with probe 4184, have all 6 of these in the third column with probe 5382, etc. Now we could do that stepwise manually, but for the purpose of this tutorial we will use the auto load feature. The first thing I'm going to do is select probe 185, and you can see that that's been selected up here. And now I'm going to click the first DNA sample, hold down the Shift key, and click the last DNA sample. Go down to the Quick Load and Run button to dot blot and use option 1, load the same probe with different DNA samples, and that automatically loads that first column. I'll take the second probe and shift click again. And I'm going to repeat this process for the remaining columns. And the last one. Now we have loaded all 6 of those columns, and the only thing we have to do to run this dot blot is to click the Run button. I'm going to make it easier to see what's in these columns. So it's kind of like a spreadsheet in that you can resize these columns. You have to do it somewhat slowly. It makes them a little bit easier to see. In cell A1, that's probe 185, the known mutation for 185 against a control DNA sample for 185. If that didn't turn dark, you'd know something was wrong with the procedure. That's a control for the 185 mutation. Notice right here, probe 4184, DNA 4184. So that's a control for the 4184 mutation. And if you go down diagonally, probe 5382 in DNA 5382, that would be this result. So you'd expect those 3 to be dark. Otherwise, again, something's wrong with the procedure. That's the way this would be interpreted.
This concludes the Southern Blot dot blot tutorial.